A very good morning to everyone, and I would like to thank the organizers for giving me this opportunity to present my work at the 6th International Cancer Conference. I'm a postdoctoral research associate at the Newcastle University Centre for Cancer, and my research focuses on understanding tumor heterogeneity following neoadjuvant chemotherapy in ovarian cancer. Ovarian cancer is one of the most lethal gynecological malignancies with a 5-year overall survival of approximately 40%, which goes down to roughly 10 to 20% at later stages of the disease when majority of the cancers are diagnosed and the cancer has already metastasized to various organs. Most ovarian cancers are of epithelial origin, of which majority are of high-grade serous type, harboring key genetic alterations like alterations in TP53, BRCA1 and BRCA2, NF1, RB1, etc., which are specific to the subtypes. The other epithelial subtypes include low-grade serous ovarian cancers, mucinous, clear cell, and endometrioid types. And each of these subtypes associate with key genetic alterations like mutations in BRAF, KRAS, NRAS, ERBB2, or commonly observed mutations like ARID1A and PIK3CA, which are very specific to these subtypes of ovarian cancers. The standard treatment for ovarian cancer comprises of cytoreductive surgery and platinum-based chemotherapy. For the late-stage ovarian cancers, however, if the patient is fit for cytoreductive surgery, then she is treated with adjuvant chemotherapy following surgery with or without bevacizumab, which is a VEGF inhibitor, and PARP inhibitor, which is a DNA damage response inhibitor, as maintenance therapy. However, increasing number of patients are being offered neoadjuvant chemotherapy at late stages of this disease, primarily due to the extent of the disease and also the poor fitness of the patient. These patients have neoadjuvant platinum-based chemotherapy followed by interval cytoreductive surgery, and the remaining cycles of platinum-based chemotherapy followed by bevacizumab or PARP inhibitor as maintenance therapy. However, as mentioned in my previous slides, late-stage ovarian cancers generally spread to multiple sites within the body, including but not limited to the omentum, the peritoneum, uterus, pelvic lymph nodes, liver, etc., which might respond differently to treatment, contributing to not only inter-patient heterogeneity but also intra-patient heterogeneity. That is, differential response to tumors within the same patient. Therefore, my research question focused on understanding how the tumor transcriptional and immune landscape changes in response to neoadjuvant chemotherapy in these late-stage cancers at different disease sites. For this study, we consented 39 women for participation in this study between 2018 and 2020, of which 23 underwent neoadjuvant chemotherapy followed by interval debulking surgery for treatment of confirmed high-grade serous ovarian cancer, which is increasingly being offered to women which are not suitable for primary cytoreductive surgery. Following sample quality control, 4 participants were excluded due to lack of enough tissue or tumor content, and we could use around 50 formalin-fixed paraffin-embedded blocks from 18 study participants with paired pre- and post-neoadjuvant chemotherapy tumor tissues for further downstream analysis. This included post-NACT tumor tissues collected from various metastatic sites like ovary, omentum, as well as the lymph nodes. These samples were analyzed by RNA sequencing, which identified significant differences not only between pre- and post-chemotherapy tissues in general, but also identified distinct differences in gene expression profile between different sites post-chemotherapy, primarily between the omentum and the ovary. The numbers that you see is the differentially expressed genes in the different subgroups when he compared it with the pre-neoadjuvant chemotherapy samples. The gene expression data was used to estimate gene expression signature scores for 10 different biological processes representing the different hallmarks of cancers. This was performed through a proprietary data analysis platform called Clarity from Almac Diagnostics. Hierarchical clustering of these gene signature scores identified 3 distinct gene clusters. Cluster 1 and 2 comprised exclusively of post-NACT tumors, and cluster 1 being primarily tumor deposits from the ovary, and cluster 2 being comprising of tumor deposits from the omentum. post-neoadjuvant chemotherapy and showing distinct differences in the gene signature score profiles. As you can see, the pattern of the red, which resembles increased expression of certain pathways, is different in the ovary, which is the yellow box, and the omentum, which is the blue box. Cluster 3 was, however, a mix of pre and post neoadjuvant chemotherapy samples with a characteristically distinct gene signature profile as compared to the other two clusters. A correlation analysis between gene signature scores identified a very strong positive correlation between genome instability signature and evading growth suppression signature, and between inflammation, immortality, and EMT signatures. The strongest negative correlations were observed between genome instability signature and immuno-oncology or inflammation gene signatures, which was an interesting observation. Further analysis of the genome instability signature showed that the average genome instability signature was significantly lower in post-NACT tumor tissues, irrespective of the metastatic site, and this was largely reflected in the individual gene signature score profiles. And this decrease in genome instability associated with increased ability of homologous recombination repair efficiency as confirmed by the measurement of the RAD51 foci shown in green here. RAD51 foci is a strong functional indicator of homologous recombination repair function involved in DNA double-strand break repair. This increased repair ability and reduced genome instability indicates selection of treatment-resistant clones following chemotherapy that are unlikely to respond to targeted therapies like PARP inhibitors as maintenance, which work better on cancers with loss of functional homologous recombination repair pathway. The analysis of the immuno-oncology gene signature associated with avoiding immune destruction, as well as tumor-promoting inflammation gene signatures, highlighted clustering of the samples into two predominant subgroups. However, interestingly, the increase in immuno-oncology gene signature was observed predominantly in the tumor deposits of the omentum and not in the ovary and other sites. To delve deeper and understand its likely impact on the tumor composition, a deconvolution algorithm called XCell was used. This algorithm uses bulk RNA-seq data to estimate the proportion of different tumor cell components while specifically focusing on different immune cell types. This approach again identified three clusters, with cluster 1 being predominantly post-NACT omental biopsies with high tumor stroma or immune score, and high proportion of effector CD8 and CD4 positive T cells. Cluster 2, on the other hand, comprised of high stromal cell population with high proportions of cancer-associated fibroblasts. And cluster 3 was characterized by low immune infiltration score. This therefore highlights very distinct site-specific differences in ovarian cancer pre and post chemotherapy. To validate the observations from in silico prediction algorithm, we analyzed the proportion of CD8 positive and CD4 positive T cell distribution in the tumors, and it was identified that the subgroups with high immune score had a significantly higher cytotoxic T cells, both CD8 and CD4. However, the increase in CD8 percentage was more statistically significant. And this increase was observed in the omentum, and not only by the XCell prediction algorithm, but also by immunohistochemical analysis. In summary, our ovarian cancers show most significant heterogeneity, not only between patients, but also between disease sites in the same individual. Gene signature analysis of the 10 hallmarks of cancer identifies distinct site-specific gene signature profiles. Genome instability gene signatures is significantly lower or reduced in post-neoadjuvant chemotherapy, which correlates with increased homologous recombination repair deficiency of these cells. And it highlights that they have a better chance of survival. Neoadjuvant chemotherapy activates immuno-oncology and immune signature, but this is only observed in the omentum and not in the other disease sites, highlighting the immunological cold nature of these tumors and the extreme heterogeneity that needs to be tackled when addressing challenges like developing new treatment options for these treatment-resistant cancers. I would like to acknowledge my mentors, Professor Nicola Curtin and Dr. Yvette Drew, for supporting me in this work, and all the lab members. my funders, Newcastle Hospital's charity, and all the women who contributed through participation in the studies. With this, I would like to end my presentation and happy to take any questions.